Hello and welcome to our walkthrough through the Rosetta tutorial on motif grafting and scaffolding. I'm just going to be working through the tutorial, and while I do this, I can explain a little bit why am I looking at something or what is interesting about it, what we're just doing, and also to just navigate some of the problems that can occur. This is actually a pretty straightforward tutorial, but it's really, you should really open up things and look at them in order to understand what actually is happening. It's like you learn the most by really understanding what's happening. And only then you'll be able to transfer to your own projects and problems that you're interested in. So there are three things you're going to need in order to execute this tutorial. The first one being Rosetta. So I'm going to check whether I have Rosetta available here. And I can just do this by typing Rosetta scripts like this. And what it does, it starts Rosetta scripts, and the only thing that Rosetta scripts is going to be returning is there's no protocol for this. And this is exactly what happens. So Rosetta is running here. The other thing I'm going to be needing is PyMol, you can use any kind of visualization tool you feel comfortable with. I'm just going to be using PyMol, and you're going to see throughout the tutorial that I'm going to point out a lot of things in PyMol, and this is how I also mostly control where the protocol works in the first place. This is obviously not an in-depth analysis, it's just more like I open this and I look at it in order to understand whether the protocol ran successfully. So let's see if PyMol is there. It is, very good. So the other thing you're going to be needing is the tutorial itself. And you probably already downloaded this, and I did so too. I am already in the scaffolding folder. And what you should see in this folder are subfolders for each of the tutorial sections. So they're going to be Sidechain grafting as the first one, then we're going to do backbone grafting, and then in the end, FunFolDes design. There is an old Rosetta remodel tutorial that is not part anymore because FunFolDes design as a method superseded it. However, if you want to use Rosetta remodel, this is a good starting point to check out how it's going. Other things you're going to find in this folder are presentation, the presentation slides, and also the tutorial itself. So I'm going to be using the Markdown version of it, but there should also be a PDF version and some other versions for your use. I'm going to open it real quick and take a look at the tutorial introduction. So this is the tutorial on motif grafting and scaffolding. I will just point out the commands I'm going to be using and some things that you need to take care of. When you work through it, you should obviously read the text and work through it. So there's a small introduction. It mostly reiterates things that are already in a presentation. But motif grafting, let's just briefly define it. Motif grafting means we are transplanting a backbone or a sidechain from one protein onto the other protein, and we're going to be using three different techniques. The first one, we are starting with the sidechain grafting. And sidechain grafting means I'm just going to transplant a sidechain from one protein onto the other one. Obviously, sidechains need to be somehow defined in space, and what's defining it in this case is what we call a context. That's the binder for the sidechains for this motif. What the tutorial does and also what the protocol does is we need to define what exactly is the sidechains we're interested in. Then we have to look at scaffolds. We have to run and actually do the motif grafting. Then we're going to briefly look into why sequence design, and looking at this or reiterating over it is really important. And selection and improvement is obviously normally a very work-intensive step, and you need a lot of inputs. So we're just going to briefly discuss this. So let's get started. In order to do so, I'm going to move into the Sidechain grafting folder. As you can see, here are already some things prepared. So we have some input files, we have a folder called scaffolds, and we have scripts prepared. There's also expected output, and we're going to come later to this, why this is important. in order to work here, I'm going to create my own folder. It's going to call my files. It's very similar to the other tutorials. This is just in order that we have a working directory where we don't overwrite input files. That would be a little bit harder to recover. So I'm going to move into this folder called my files. So the next thing is in the tutorial is that we're going to be needing our input files. And that is mostly the motif and the context as it is defined by a co-crystal structure. Obviously, normally you would go to the PDB and download it there. And you would also have to do a lot of like defining and what is actually your residues that are interacting. Obviously, in this kind of tutorial, this has already been done. So we basically already know what we're going to be doing. However, I'm going to download it from the PDB nevertheless to show you how you can clean it up. So in order to do so, I'm just going to use a script called clean PDB. It's provided inside Rosetta in the tools directory, and it's really useful in the script that you can use to clean your files and to clean up and isolate chains, something like this from your PDB files. The protein we're going to be working on is the estrogen receptor bound with a little co-activator peptide. And so the things we're going to be needing is as context the estrogen receptor and as motif this peptide. The PDB we are working with is 1GWQ, and there in chain A there's a copy of the estrogen receptor, and the respective binding peptide is chain C. So in order to isolate these, I'm gonna use this very script that I just mentioned to you. So I have to find my Rosetta copy, and instead of going into main, I'm gonna go into tools, Grafting tools, scripts, and there's a lot of useful scripts, but one of them being clean PDB. And I'm just typing, I want to clean up 1GWQ and I want to clean up chain A. What the script returns is it doesn't find 1GWQ in my folder, which you could obviously have copied from the input folder too. But as I can't find it, it's going to download it from the internet. Obviously, you need to have an internet connection for this. What you can see, it's isolating chain A, and this is the fast that it has been outputting. So I'm going to do the same thing for chain C. This should be the peptide. And you can really see the peptide is really short. So this is what we expect. In order to maintain the nomenclature that we use in this tutorial, I'm going to rename both of these files. And 1JWQA PDB. This will be our context. And the other file, 1GWQC, this will be the motif PDB. So, context motif, this looks good. Kind of as I said, you can do this whole totally blindfolded to just execute the commands, but it's definitely going to be a lot more interesting if you look at these things. So I'm just going to open them in PyMol, the context and the motif. So we are kind of zoomed in onto the motif. So here you can see one of the copies of this dimer of this estrogen receptor and the peptide. And now we kind of want to see the interacting residues. So that's going to be interesting going forward. So if you just make this stick, you can see that here are these two leucines that are really interacting with the protein interface. So if I would have to put down my bets on if these residues are going to be important, I think I will win. Remember these two residues as we move forward. We're going to come back to them over and over. How it looks like, we have a motif and we have a context. And this was the first subsection we needed to do in our tutorial. Let's check out what the next thing is we need to do. So we downloaded everything. So the next step is preparing a scaffold database. We already provide a scaffold database here in the tutorial because it's relatively work intensive and we need sometimes also different software in order to sample that. You can also obviously do this very manually. There are some guidelines and sometimes it's a little bit defined from your project on how you want to select scaffolds. And you're going to find a selection that we just provided you with in the scaffold folder. So we are actually not going to be working through these things. However, keep in mind that if you do that, you need to clean up the structures. There are also like Rosetta grafting, it could only be a single chain protein with a ligand, without ligands, and without like any weird things in there. The next thing is, if you really want to use them, you should relax these things beforehand. Same is actually true for our inputs. For the sake of time, we're actually not going to do this. But head on into our next section. So this is Motif Grafting and interface design. So the things we need to do here is we need to copy our input files, which would be Motif XML. And we're going to look at this. The other thing is we're going to connect our scaffold library in a list so that we can point Rosetta to where it is. And then in the last step, we will have to run the protocol and check whether it's running successfully and what's happening. As you can see, we're going to run the protocol only with a single output for each Motif graft attempt. This is for the sake of time. If you run these things, you should always create a lot more than we do in the tutorials. If you're doing that, you should actually use the multi-post mover. But you can check that out at the documentation when it comes to that. So let's go ahead. So first thing we're going to be doing, we copy from the scripts folder the Motif Graft Sidechain XML. So that looks good, it's here. The next thing is we need to prepare the list with our scaffolds. So the scaffolds are in the scaffold folder that is also part of the files we provide. You want to maybe put a star PDB. So now we have all the files here. Let's take a brief look at the scaffold library we provided you with, because this is actually interesting. Your scaffolds are actually like an integer part of these protocols. Selection can really make or break it. If you have good scaffolds, you're gonna find something you can graft on. If that is not well prepared, it might be very hard to find a matching scaffold. However, there are options to run, like to iterate over the whole PDB and find scaffolds. Let's just align them onto one. So this is what you've seen already in the presentation. These are all scaffolds that we provided with. This is a pretty big scaffold. I tend to use smaller ones because I think it's easier to see. This is still pretty big. This one is tiny, but might be very useful. As you've seen, the peptide we'll be using is very helical. So I could imagine the chance of grafting it onto something like this is actually not that bad. But yes, we have a lot of things we can try to graft on. And this one also looks pretty nice. So we're going to see which one of these works or doesn't work. So the other thing we have here now is the Motif Graft Sidechain XML. So I'm just going to show you that. It is obviously a Rosetta scripts protocol. We have the protocol section here. The heart and core of the sidechain grafting protocol and also the backbone grafting protocol is the Motif Graft mover. You can see that inside the Motif Graft mover, you have to define inputs, for example, the context structure as a context PDB, the motif structure, which we named motif PDB, and then we have predefined C tolerances, which I don't necessarily touch on. There's also some testing that the protocol does. And this is really interesting. So in sidechain grafting, you have to define which are the residues that needs to be transplanted, and they are called hotspots. And in this little option, you define which one of these are the hotspots. And here it says 3 and 7. So we're going to check out if 3 and 7 is correctly. If you put the wrong one there, you might end up with something you don't want to have. So there are some further things that you have where the protocol is told like how much of the motif we can actually leave out and these things. I really would advise to start playing with this if you're working on your own project and you have a use case. Very important for sidechain grafting are these options. Full motif backbone alignment set to true, because yes, we want to align the full backbone. Graft only hotspots by replacement set to true. This is actually what tells the protocol that we're only interested in these hotspot residues, which are 3 and 7 at this point. Revert graft to native sequence, yes, for everything that's not a hotspot. So this really is a sidechain grafting protocol, and it looks like everything is set up correctly. Obviously, there's some post-processing and processing in there, which we won't go into detail here. So I'm going to run this now, pointing to Rosetta. Rosetta scripts to be true. So we have to pass the protocol. Oh, I should type this correctly. We also have to provide the scaffold list, because this is how Rosetta is told where the scaffolds are to be found. Obviously, if you can put into this list, then your chosen library. The X1 and X2 are options for rotamer something, and this is a default that I would also advise to keep. It looks like we are pretty done on this, and I'm just going to hit enter. And so you can see Rosetta scripts is reading in first here the scaffold library, and it's also reading in the XML, and it's been passing the XML section, so it's running now. Here it's actually going into the Motif Graft mover, and you can see how it's trying to find a graft position and how it's testing some things. And I think there's a point where it actually says that here you can kind of see that it's now actually executing the protocol and finding some positions, and here it's already in a post-processing. However, it's going to take some minutes, and because we don't want to wait for Rosetta, we have some prepared output that I will show you now in order to discuss what we expect to happen. So if you go back into, I don't even know where I'm here. Okay, this is the wrong folder. I'm going to go into Sidechain Grafting. You can see that there's an expected output, and I'm just going to go in there and discuss with you how we expect the output to look like. So you can see a lot of PDBs in here, and not all of them are output. So ignore this one. This is input for the next step. But I would just pull up everything that has a 001.pdb in the end. And then I'm also going to pull up, just for comparison of my files, Motif PDB and the context PDB. And then we're going to check out whether this output that is pre-generated but available actually contains some sidechain grafts. So this is what we see. I'm going to color everything gray. And then we're going to make this motif green again. And the context magenta. So just a reminder, this is the context. This is our protein of interest, in this case, the estrogen receptor. And this is this co-activated peptide that was bound. And we're having these two side chains. We're mostly interested in number 3 and number 7. And if you look at them, they are in the interface and interacting with the receptor. So we expect in the sidechain graph these two residues to be grafted. So I'm gonna, this is a pretty big one. However, when you look at this, it's overlaying correctly in this position. And let's check out if we're gonna find alanines at leucines at the right position. I think I'm a little wrong here. No, I'm not so wrong here. Oh, there's a second one. Yes. So, as you can see, you're going to have leucines in this position in your scaffold. So although this has been a very big protein, you can see that you successfully grafted the side chain and they maintain the interface as we predefined it in the complex between context and motif. Let's see if there's a little one. Yeah, this is a little easier to see. So this tiny protein, let's see if it also contains leucines at the right position. And that is the case. So this looks actually super nice. You can even see that this portion really captures it all very well. This looks like a really good sidechain graft. Obviously, there are a lot of things that you need to look at. You have to sample a lot of these designs. And there's something happening. The protocol is allowed to adjust because of the insertion of these residues and redesigns parts. Not always are all of the residues it adds are really useful. And there's this rule of thumb that the more you mutate on like an existing protein, the harder it's going to be to predict if this protein will fold correctly and behave correctly experimentally. So in order to propose sequences that you actually want to produce, I would definitely go back and check every single mutation, whether it was necessary or not. And in the next step in our tutorial, we're going to exactly do that and look at how much is actually necessary for one of these examples. So I'm just going to close this. Let's check out in the meantime how far sidechain graft actually moved it to. You can see it's still running, but it has been running now for a while, so I bet we already have some outputs. Still, as we are still waiting on this, I'm going to go ahead in the tutorial and do this very next step, which is called selection and improvement of designs. This is where we look into if Rosetta changed something and I reverted back, was this actually something that we need in order to maintain structure and score? Or is this something that was added in, but it's actually not necessary to change that? And this will allow you to differentiate between mutations that you want to keep and mutations that you might not want to integrate in your designs. So what's happening here is that we're going to combine the context PDB with the scaffold protein and make a complex out of it. Obviously, this would not be binding correctly. And then we're going to compare it to our design, and every amino acid basically that's different, we're going to check out whether it was actually necessary for the score. So I'm going to execute this in this window. So let's move back into my files and get this started. So we're gonna combine context PDB and the scaffold 1JIQ to native complex is how we call it. And now we're going to run a command in order to compare the mutations between them. Source bin. And the application we're using is called Revert Design to Native. There are some options that need to be parsed. So we have to tell what one is the wire type. That will be native complex. And we're gonna pass the output to file so that I can always look at it again later on and it's not going away. So I'm just gonna start this in the background and it's gonna write the output to this revert log. So we're still running sidechain grafting. I'm gonna go into another sidechain grafting. Let's see how many of the files are already correctly produced. So we are quite at the end of it. So sidechain grafting is actually not that slow because the confirmation changes needs to sample are a lot more restricted than other protocols. It actually is quite fast and allows you to sample a lot of scaffolds in a relatively short amount of time. So both of them are still running, but this should be very much at the end. Design is normally like one of the most, just a little bit more work intensive. Now it's grafting again. Yes, as this is still taking some time, I'm gonna start the next section of the tutorial in another tab. So the next step, what we want to do is backbone grafting. Backbone grafting uses in a lot of ways the same things. It also needs a defined context, a defined motive, and it needs an XML as an input. The XML in a lot of cases is very similar to the sidechain grafting XML. There's a little bit different options and a little bit different thresholds. Which also tells you it's sometimes very important to look at the options and thresholds. Once we get this running, we're gonna go back and look at these outputs from sidechain grafting and where we check the designs. So let me move into this tab and then to the part of the protocol that's called backbone grafting. When we check out this folder, it's very similar to the other one. We have input files, we have a scaffold library, which I think is a little different than the library we provided. Is it actually? Yeah, I think it's a little smaller library than we provided for sidechain grafting, and also a little bit different examples. So again, we're gonna make a folder that's called My Files to not mess with any of the inputs. So I'm gonna make a folder real quick and move in there. So now I'm gonna collect the input files. I'm gonna copy from input file order motive. I'm gonna copy from the input file folder context. And as I'm already doing this, I'm gonna go ahead and also copy from scripts the Motif XML and copy it here. So the last thing we're going to be needing is the scaffold library, and we're just going to do it as we did it before and just create a list with our available scaffolds. Let's check scaffolds. This looks good, it's pointing to the right direction. The other thing we want to check out is if motive and context are looking good. It should be this exact same example as we used before. So we again have the estrogen receptor and our small peptide to find this motive, as you've already seen it before. This time we are not only interested in grafting these tiny two interacting residues, but we want to graft this whole thing. So how is the motive graft mover achieving this? So we can look at the XML. Overall, it looks very similar to the Sidechain Grafting XML. We have here the Motif Graft mover. Most of the things are very similar. Context PDB needs to be defined. Motif PDB needs to be defined. However, RMSD tolerances are a little different, like higher thresholds, which allows you to incorporate scaffolds, which are less similar. We still have to define our hotspots as residues that aren't allowed to be in any way exchanged. These are options on how to sample the motif space. This option is very important for backbone grafting because full motif backbone alignment is set to 0, which means we are not looking for things that are tightly aligning with our motif backbone. Here we have graft only hotspots by replacement set to 0 true. This is the option that says like, please maintain the backbone residues. As backbone grafting also takes some time, I'm just going to start this and then we're going to go back and look at some of our output from sidechain grafting and see if this has finished and what the results are from revert our design. The command is also very similar to how we used it in sidechain grafting. We're going to input our list of scaffolds. Am I missing something? Instruct 1. This is important. However, I think this will do. Rosetta will tell us. So I'm just going to hit start. It's reading in the scaffolds. It's reading in the XML. And now we are running. Perfect. Yeah, so you can see it already reported for the first graft, some results here. So we're going to keep this running and go back to the sidechain grafting and the revert to native. So this is our sidechain grafting. As you can see, this finished successfully. I'm going to check this out. Yes, we have a bunch of designs here. You should go ahead and look at your results. What is very important, it might be that your results, the results in the expected output folder, which we looked at already, might be a little different to each other. That wouldn't surprise me at all. The other thing we wanted to check out is what happened to our revert design, like revert the design to native, and whether we actually needed all the mutations we made to the scaffold. So I'm just gonna cut the log file. Yes, and this is what you kind of see. We are mostly interested in this reporting in the end, and this is why we actually wrote it to a file. You can see how it changed the residues we were interested in and reported the scores for it. And here in the end, it can basically say which one of the mutations you can just ignore because they were introduced at a point. Either they didn't change the score very much, or they were just there to overcome like a local energy minimum, and you don't actually need them. There are, on the other hand, also some mutations that you want to keep. And this is mostly to really fulfill this rule of thumbs that you should introduce as few mutations as possible. And if I would have to select a sequence, I would always be relatively conservative about this. Cool, so we just finished the sidechain grafting. I'm probably thinking that backbone grafting is still running. You can check that out. Yes, this is true. In order to keep it short, we're going to look at the expected outcome from backbone grafting that we provide with the tutorial. So I'm going to go into this tab again and move to backbone grafting. So you see we are already generating output. So in order, I'm actually going to take our output instead of taking the prepared output. And I'm going to take all the PDB files in here and open them in PyMol in order to look at them. This is a zoom in on the motif region. Let's zoom out a bit. So I'm going to make this big, and again, I'm just going to color everything gray. And this I'm going to do green. And this I'm going to do magenta. So, just as a reminder, we are still working with this estrogen receptor structure where we have this one copy of the dimer bound to this peptide with these interacting residues here. And you saw in sidechain grafting how we transferred these two residues into helical stretches mostly. Here we expect like this part to be grafted, but we're not so much restricted on what kind of backbone. So it's going to be interesting to see how this is going to look like. So let's open this first one. Wait, this looks a little wild. So something that happened here, I could imagine that happened here is you can see that this overlaps. So, and something you can also see is like the context is here. So it's always done in the presence of the context. Something that happened here is probably it was grafted because it found something in matching backbone. But when it post-processed it, the interface blew up and the proteins were basically more happy to be separated than they were happy to be together, which is always a bad sign for the graft. It means that something is relatively fundamentally wrong. And I could just imagine that there's a big clash or something that moves them apart. So this is probably something you don't want to move forward. So let's check out this one. This really looks very sweet. You can see how this backbone really aligns very well. And just to compare, I think I'm gonna just load from our scaffolds folder the backbone of our input structure. And I'm going to align it to our graft. So you can see the difference in the backbone here. Okay, this looks really cool, like a very successful graft. So this is the backbone of the protein in this region, which you see is very different from being helical. This is the motif you're interested in, and if you align this, this doesn't align very well in this section at all. Like if you would revert some residues here, you would never get the interactions you wanted. However, what you can see is like that these N and C termini are aligning relatively well, and this is what the protocol looks for. So when you compare these now after the graft, you really successfully transplanted a backbone, and here the protein didn't move out and is still very much aligned with this interface, which is an indicator that the interface is not disrupted. So this one looks also pretty good. You can see that this has also been very successfully transferred. Yeah, this one moved a little bit. This is a bigger protein. Sometimes it's very beneficial to have more interface because during design you can also create favorable interactions. However, if you want to keep it simple, I really would check that the interface really aligns very well with each other. So compared to something like this one, it's not too different, but probably the spar one would be my favorite. So yeah, this is how the output of backbone grafting looks like. And while our backbone grafting is still running, it takes a little bit more time than sidechain grafting. Or maybe it's already done. Oh, it is already done. So we finished backbone grafting, and you can see that we generated some structures based on our templates. Did we actually generate output for every template? It looks like it almost. So this was a very well selected scaffold library. So sometimes, or even if you select scaffolds very carefully, it might be that it just rejects the scaffold because the thresholds are just not reached. This also brings us to the next step in our tutorial, the next and last step. And this is functional folding and design, FunFolDes, which is a pretty new protocol and really exciting because it's super powerful. As you've seen, the scaffold really brings a lot onto the table in these protocols, and you are really both restricted and guided by the scaffold. There are definitely a lot of epitopes that this is undesirably, or you won't find a scaffold which you can use for this kind of epitopes. So you really want to have protocols at hand that you can use for epitopes that are a little bit more complex than what we've looked at so far. So I'm going to be moving into the FunFolDes folder. Which is also prepared very similarly to these other folders. We have some input files and output files. FunFolDes needs a lot more time than both backbone grafting or sidechain grafting, which you can finish in time in our tutorial. For FunFolDes, you're running half an hour or something for one output structure. It also is due to the fact because it's doing a lot on the protocol and sampling a lot of conformational space. So I'm gonna check out the tutorial on what we need to do. So we've done backbone grafting. I'm moving to FunFolDes. Again, we will have to prepare folders for our files, for our working directory. We have another example that we're going to be using here. So it wouldn't be much fun using a very small helical stretch because it's easy to find scaffolds for that. The example we are using in FunFolDes is a little bit more advanced. It's the epitope of the antibody moterizumab, and it's structured a little bit more complex. It contains a loop, for example. It would be really hard to find scaffolds for loops, just because there's so many options on how they could be oriented. And so one of these input files is the co-crystal structure of the antibody with the epitope. The other input structure is a selected scaffold. Obviously, in order to run FunFolDes, you would need to select scaffolds beforehand too, as you did for sidechain grafting and backbone grafting, and we discussed both in the appendix of this tutorial and in our presentations of options for that. However, this is the scaffold that is known that this protocol will work well with this epitope. It is actually reported in a publication. So this is why we're taking this example. What we need is also the input files, both context motif and scaffold, and then we're going to be needing our XML. The other thing we're going to be needing will come when we look at the protocol, and the final thing we're going to be doing is we will run Rosetta using FunFolDes design, and after that we'll be looking at some of our designs and check out how this protocol performed. So I'm going to move this over and start out in this folder and make a directory. Copy over our input structures from our input files directory. There should be 1kx3. This comes here. And this too. And our input protocol. So this looks great. We're going to check out the structure because they're a little different than the ones we used before. So it's good to orient ourselves. So this is pretty big. However, this is just our context. This is the fab fragment of the antibody moterizumab. You see here the complementary determining region with the CDRs, CDR3, interacting with this small epitope that has been co-crystallized with it. And you can see, although it contains secondary structure elements that we might actually be able to find something for, it's relatively hard to find something for these loops. And as scaffolds are scarce, we might not find something that has the right RMSD to use one of the previously discussed protocols. Especially look at the scaffold, like overall structurally, this region that we aligned here is very similar. So you again have helical stretches and a loop connecting, but you can see that this is an RMSD that would be during backbone grafting or sidechain grafting, just discarded. So although this topology looks good, we can't use any of the grafting approaches, and this is where FunFolD design comes into place. So what FunFolD design will be doing is it's going to keep this motif here and in context with this binder, and it's going to take this topology, constrain it using coordinate constraints, and refold the protein into this topology. However, during folding, there's a lot of interactions that are going to be made also with the motif, which is can't move, can't change, can't design, can only repack, so that the backbone of the scaffold really starts supporting your motif. And this can really overcome this high barrier that you have when you keep your motif very static, such as sidechain grafting and backbone grafting did. So let's check out how the protocol looks like. So something that you can already appreciate, the protocol is a long, a really big Rosetta scripts protocol. It's really commented for your purpose, so you can like differentiate between all of the different sections. And we're gonna go just walk through it from the top to the bottom. So obviously here score functions. This is a very, very important part in running FunFolD design. You remember that in Motif Graft Mover, we defined what is the context, what is the hotspots, what is my motif. And in this case, we won't be using a similar syntax. Here in this protocol, residue selectors define what is motif, what is template. And this whole section that I just marked is really where it's also telling the protocol which things are grouped together for which purpose of the protocol. So you could imagine we will allow hotspots not to be designed. However, we will allow the we will also not allow the context to be redesigned because this is our binder. But we will allow to redesign non-hotspot residues in a motif and scaffold or template in this case residues. So we're going to make selections of these groups in order to define what is allowed to happen there. So this is happening in this section. If you ever have to adjust this for your purposes, when you're running with your example or your project, this would be happening in this region. Like it's indicated that template, this is our scaffold protein. We have to define the acceptor point where we want to insert it. We have to define what is the motif, and so forth. Then there's also some things that's defining how and what is allowed to move, how much. And it's also a part that tells Rosetta what it's allowed to do with which kind of residues. Kind of just what I said, it's like, it's not going to be allowed to be redesigning context and hotspots, for example. Then there's some filtering options that will be used in the backend of the protocol in order to generate both scores and also tell you something about how it's been run. So this is how the protocol moves, and this is actually also in the right order. This is more technical things, but really interesting for you is you can see in the pre-processing, there's a heavy constraint generator. This is how we define the topology. And this is really hard and core of this tutorial. This is the noob initial mover, which is called FunFolD design here. This is really the part, and noob actually kind of refers to having like a something in protein that you can't change, which is our motif. And initio means that we are doing basically ab initio folding, and this is also why we need fragments for our tutorial. If you've ever run one of our folding tutorials, you need to provide fragments of the secondary structure in order to provide for the folding algorithm a template basis. The fragments are generated in this case not just from the PDB or something, they are solely based on the scaffold protein, which really allows Rosetta to incorporate aspects of how the scaffold protein topology looked like. Then there's a lot of clearing and post-processing, like you're gonna get rid of your constraints. This is also very important. You're gonna redesign large parts of the protein using fast design, which assembles a lot of different amino acids then in positions that are allowed to be designed. And then it's gonna, clean everything up and provide you with an output, hopefully. So something that I wanted to point out is here, especially in the fragment section and here in this section. So you find these little signs. This is the scripts variable. I think you've heard that in the Rosetta scripts talk. You don't always have to hard code your input as we did during sidechain grafting and backbone grafting. You can also pass input as a variable. In this case, our variable is called donor. So when we start Rosetta scripts, we have to define what we actually mean with donor by passing a Rosetta scripts variable. And you're going to be seeing how this looks like when I type the command. I think this is a really, really useful thing. So as input, we need our scaffold structure. In this case, we say nstruct 5. As this is an ab initio folding and really samples a huge conformational and sequence space, you should really generate a lot of outputs. So not just 5, I'm more thinking about 5,000, 10,000, so forth. This is how we pass the variables using parser script bars as the flag. Then we have to define in each of these variables. So I'm going to tell that donor is our co-complex structure. And then I have to define the fragments. So Rosetta nowadays can pick fragments on the fly. However, as this is time intensive, for the sake of the tutorial, we provide you with the fragments. However, there's a version of the protocol in the supplement that automatically picks fragments. And if you run this yourself in your projects, you probably want to take that option because it's just taking some preparation steps of you. Yes, so this looks good. This should contain everything we need. We have some ignore flags. They are just more precautionary because if you download a PDB from the protein data bank, there are going to be some things that Rosetta doesn't really know what to do with, or you actually don't want to do anything with it. So you just basically tell it to ignore them. So something went wrong here. Just checking my input. Okay, I'm going to hit start and see if this runs. Okay, I have a typo. Parser script bars. So I have a typo. I have more typos. Frags, frag 3. Oh, no, just incorrect. Oh, I know what the problem is. So this was good, but this wasn't good. So now the protocol started running. It passed our input, our scaffold, and it also passed the protocol, which Rosetta successfully read in, and now it's running. Oh no, it's not running. I did something wrong. What did I do wrong? I think I have a typo again. Case 8. So what is going wrong? Okay, I think I have the wrong name for my script variables. Let's check it out again. So they're called frax9 and frax3. Frax9, frax3. $203 miles. Where is the problem? Oh, I know what the problem is. I didn't copy the fragments. So easy. So I'm telling Rosetta to read in these fragments and it can't find them, so it crashes. So let me copy the fragments from our input folder. And restart this. This looks good. And now it's running. Yeah, this is how it should be looking like. As I already said, FunFolDes design is pretty heavy and it needs a lot of time because it's refolding the protein and sampling a lot. You'll probably have to wait like half an hour for your first output. To make this a little faster, we will switch and look at our our expected output files. So if you go into the FunFolDes design folder, there's output files. And this is where we provided you with like 30 example outputs. So you can actually check out how it's looking like when the output is created. So I'm going to open them in PyMol. And I'm also going to open up our two input files. So we can compare input and output. And you're going to see a little bit of a difference compared to sidechain and backbone grafting. Let me move this here. We do the same as before. We're going to color everything gray. And then we're going to color our motif and binder green. And this is our input scaffold, which I'm going to color blue. This is just to compare how the native scaffold looked like with the final example. Something that you can already see from like these 30 outputs, which have not been really much scored at all. Or like we really ran 30 and gave you 30. Something that you can already appreciate is that the motif section has been like, this has been rigid and very much defined by your protocol. So there's nothing that changed very much here. The blue thing that looks different is the scaffold. You can see everything that's gray and green perfectly overlays. This is the motif region. But you can see how Rosetta really sampled a huge space and a lot of options on how to like use the scaffold in order to stabilize this motif. So let's go through some of these example outputs and compare how they look like to the scaffold. Okay, this is the first one, and you can see how this helix really moved here in order to incorporate the motif. So this is a relatively drastic change. Oh, this one actually doesn't look so good. You see how this really unfolds. This is something that I would probably discard. Again, this one moved a lot. This looks better. You can see how the scaffold maintained a lot of its placement of the helices. However, and this is also to compare to the protocols we looked at before that the scaffold was very rigid and couldn't really adjust for anything. You can see how much this can adjust. So sometimes some of these changes might actually exactly what we are looking for. The other thing is also, I don't know if you can see that very well here. We have been redesigning this sequence very drastically. So it could be that the sequence you're getting out from FunFolDes Design does not have very much similarity to the sequence you put in as the scaffold. Because obviously there's another sequence option for the protein fold, but it's definitely a lot harder to evaluate on is this a good result or not. So I would really highly advise you to look at the scores and look at the structure. Also maybe at honesty to native scaffold, to look at incorporation and maybe do some forward folding. And I would really recommend sampling and analyzing a lot on these outputs, because it's a very powerful tool that samples a lot of conformation space, but you also need to really select down what will likely be a good option for your purpose and for your project. So I'm going to close this and I'm going to very briefly just look into the provided score file. This is how the score file looks like, as this is not very easily to be read. So in the first line, you can always see what has been put out here. There's a lot of options that FunFolDes Design really gives you, and you can look into this. It definitely plots some RMSD during the protocol. It also reports energies from Rosetta. A first good start is always looking at the total score. So I'm normally using, you can just use arg to plot this or to extract this. This is obviously not the right column. Let's check out column 35. So we are just outputting the columns, and you can see what the total score is for each of these models. And you kind of can see that they're adopting a range. It seems to be that 1600 is a good one. There are some that are really low, and you might want to check out why that is. Oh, I think I remember why this one was low. You remember that this one was the one I said this looks a little unfolded. So based on the energy score, we would also probably discard this. And I would probably check out the best scoring options here. This one looks, for example, really good. We could check if this is a good. Which one was that? Oh, 9. If this looks good compared to our input. Oh, this is wrong. Oh, I'm always pointing to the wrong directory. Yeah, this is what we want to point to. So this looks like a good example. Oh yeah, you can really see how nicely packed this protein looks like. Packing is, for example, is something you can also look at. So it doesn't look like there's much space in the middle of the protein. It really supports the motif. And when we compare this to our input, you can also see, although it moved these secondary structure elements, it really maintained the protein structure in itself. So this looks like a promising design. Okay, with this, let's check for the final time. Our FunFolDes design is still running. I think it should already have outputted one. It's running past design at the moment, so it's redesigning the sequence. And you can see here a column of the residues, and here it's not allowed to design this, so I think this is probably the context, like our binder. So with this, we are at the end of the tutorial. I hope this was informative for you and it helps you walk through this yourself and also points a little bit out what I would be checking out when I would be doing this. And I hope that you have fun using Rosetta, and I also hope that you enjoy working with it in your projects, and good luck with that. And with this, we are at the end, and if you have any questions, you can always reach out to us. We love to help you with your projects. And I wish you all the best.